Hi, a very good day to Dr. Harry and everyone. Today my presentation is about Plasmodium knowlesi in Southeast Asia and proteins that involve in its erythrocyte invasion. So for my review paper, the content will be introduction, Plasmodium knowlesi and its life cycle, landscape factor and human infection, overview of Plasmodium erythrocyte invasion, molecules that involve in Plasmodium knowlesi erythrocyte invasion, which is include normocyte binding protein, Duffy binding protein, apical membrane antigen 1, and calcium signaling. And lastly is conclusion. So for the first one, Plasmodium knowlesi and its life cycle, I will explain about the life cycle of knowlesi from vector infecting human host being taken up again by the vector. And I will state the symptom of Plasmodium knowlesi infection, which is include dominant, common associate, and additional symptom. So the life cycle will be starting by the mosquito injecting sporozoid into the human. A sporozoid will move into liver cell, and at liver cell, sporozoid will turn into a lot of merozoid. And merozoid will go into the blood stage, where the merozoid will invade human red blood cell. And while some of the merozoid invade red blood cell, some of them will turn into gametocyte. So gametocyte will be taken up again by the mosquito vector, and inside the mosquito, gametocyte will form zygote, and then to ookinete, oocyst, and then to sporozoid. And then sporozoid will be transferred again to human host, and this is how the whole cycle will be repeated. And landscape factor and human infection, I was stressing about how forest and environmental factor play an important roles of Plasmodium knowlesi transmission. For example, in Cambodia, Plasmodium knowlesi was detected in 2 patients of Pailin province during 2007 to 2010. And in Singapore, all cases will include military personnel who took training in the forested area. In 2014, a study in South Kalimantan, Indonesia, which Plasmodium malaria was observed in the worker who visited a new mining location in a forest. Next, for erythrocyte invasion of merozoite, the erythrocyte invasion begins by the egress of infected red blood cell. Egress, which means the merozoite being released from the infected red blood cell, and priming process to invade a new red blood cell, which cause exposure of this merozoite in the environment, which is not in the red blood cell, are exposed to low potassium level. And this triggered calcium release and activated adhesin and invasive. So there's also initial interaction between merozoite and red blood cell, which is known as irreversible attachment. And RON2 protein will acting as an anchor with AMA1 protein, and then release of rupture bulk, which then parasite to forest vacuole and invasion merozoite happen. So this is the diagram that I draw, which is include invasion steps and mechanism signal. They're totally like 5 invasion steps, which is egress, attachment, reorientation, invasion, and post-invasion. So a mechanical signal overview is stated to show the key event happen in each invasion step. So before the attachment of merozoite to erythrocyte, egress happened when merozoite released out from the ruptured erythrocyte. Then merozoite are attached to new host erythrocyte and reorienting the apical end to the host membrane. In this step, irreversible attachment happens with the formation of moving junction MJ. Invasion then takes place with the formation of peripherous vacuole PV. Once the merozoite enter the erythrocyte, sealing happens at the posterior end, and merozoite is now in the PV and the red blood cell. So protein that involved in Plasmodium knowlesi erythrocyte invasion, there are 3. First one is normocyte binding protein, Duffy binding protein, and apical membrane antigen. So for normocyte binding protein, this protein is the member of Plasmodium knowlesi reticulocyte binding like family that express within the micronames. The RBL is an important parasite adhesin that is expressed at the apical end of parasite and found to be involved in merozoite invasion and host erythrocyte receptor binding. So PKNBPX alpha and PKNBPX beta are two normocyte binding protein and the only two RBL invasion ligands that is expressed by knowlesi merozoite. PK and BPX alpha protein are strongly bind to human erythrocyte, meanwhile the beta did not. The beta only bind to the monkey erythrocyte. So severe impaired merozoite invasion has been observed when the beta gene is disrupted, hence impeded the in vitro growth of merozoite in human erythrocyte. So this has shown the crucial role of PK and BPX alpha protein in human erythrocyte invasion, especially during the establishment of moving junction between human erythrocyte and the merozoite. So for Duffy binding protein region 2, the Duffy binding protein alpha is one of the protein in the erythrocyte binding light family of Plasmodium knowlesi, EBL. The EBL family is another adhesive family that is interact with the receptor of host erythrocyte that are localized in microname and neck of Erythrocytes. PKDBP alpha interact with Duffy antigen receptor for chemokines to invade human erythrocyte. The deletion of the gene that encode PKDBP has shown the importance of this protein for human erythrocyte invasion. So this protein can be divided into several regions. Meanwhile, region 2, which is rich with N-terminal cytine, contain crucial Duffy binding light ligand domain for erythrocyte binding. So this protein knockout mutant have failed to establish a junction with human erythrocyte and completely impaired the invasion. So it has been observed that antibodies that raise against this protein could terminate the invasion of human and macaque erythrocyte in vitro, making it is a possible target vaccine candidate against knowlesi malaria. So for apical membrane antigen 1, the recognition of host erythrocyte and the invasion process of merozoites are dependent on the parasite surface molecule, such as apical membrane antigen 1, so this protein. EMA1 is localized at Rhabdothrinax organelle and is commonly found in apicopexide parasite, such as Toxoplasma parasite and Plasmodium parasite. So disruption of this protein gene has impaired the merozoite ability to invade host erythrocyte, hence validate the assumption of the importance of EMA1 protein during the host invasion. Purifying selection was found strongest in the domain 2 of PKMA1 gene. This finding suggests the domain 2 of PKMA1 may be under functional constraint. The presence of purifying selection and low nucleotide diversity in domain 2 indicate that this protein should be further studied for vaccine development target. The next one is calcium signaling during invasion. So calcium ion are secondary messenger molecules in the signal transduction pathway. Hemostasis maintenance of calcium is important for the parasite survival as calcium plays an important role in the invasion, growth, and mortality of the merozoite. So when merozoites are released from infected erythrocyte during egress, they are exposed to low potassium level in the environment. So the exposure of merozoite to a low potassium level in environment has activated the phospholipase C PLC pathway through an unknown mechanism. This then leads to the inositol triphosphate production and the activation of IP3 receptor getter channel, then leads to the release of calcium from the endoplasmic reticulum of merozoite. Microname proteins such as erythrocyte binding antigen and EMA1 is released to the merozoite surface, resulted from the rise of cytosolic calcium level. Following this, the EBA then binds to glycophorin A and leads to the restoration of basal calcium level in the merozoite. So once the calcium has been restored, the Rhabdothrine proteins are released through an unknown mechanism for the invasion. So for my review paper, there's overall 45 references. However, I only put several in this presentation. So that's all from me. Thank you.